David, quando vuoi cominciamo. Quando vuoi tu. No.
Oh. I think we can start. Then we finish at 12.
12, yes.
Which question?
Yeah.
So hopefully it will be a lot.
Aspetto solo il via libera. Ok, cambio di schermo allora. Grazie. So yesterday we did a very, very brief introduction to general concept in microbiology, right? Today we'll dive into metabolism, because at the end of the day, you don't really care how microbes interact, how microbes grow, how microbes duplicate, what's their molecular machinery. What you're interested in is the interaction of microbes with chemicals, especially hydrogen, and a number of other compounds that might be related to underground hydrogen storage, natural hydrogen, and things like that. Before we start to speak about microbial metabolism, it is the very short recap of yesterday. So microorganisms are unicellular, small, abundant, and generally structurally simple organisms. Most of them are bacteria and archaea. Why did I write 4 domains of life? 2 of the 3 domains of life, sorry about that. The third being eukarya, which it's us, basically, and all the animals and all the plants. Their size can vary a lot, but on average, they are about 1 micrometer long. They are incredibly diverse, far more diverse genetically and metabolically than all plants and animals put together. I was showing you yesterday that on the tree of life, as currently we know it now, in a single line, you find all diversity you think about, and you can think about, and then there are hundreds of other lines that contain as much, if not more, diversity. We can culture them, but we can culture only a small fraction of the microbes out there. So we don't really know what they do in most cases. And very often, we don't know the gene function. So we know there's a gene there, we can sequence it, we can read the sequence of nucleobases. So we're able to read the language, we just don't know what it means. That's what basically is. Today, we'll dive into basic metabolism. I've tried to simplify, yes. No, anytime.
Gene sequence. How do you know, like, when and where genes start and where it ends from, like, with people?
That's a very good question. After analyzing a lot of genomes and after manually testing what portion of a gene is transcribed into what we call the RNA, which is the working copy of information, and then what portion of that is actually translated into a protein, we've been able to figure out that there are only a few combination of letters of nucleobases. So the way DNA works is that there are 2 long chain of phosphates that are bound to each other, and then there is a series of only 4 chemical structures that can happen inside. And these 4 chemical structures are paired 2 by 2, and they're paired based on specific chemical bonding, like 2 hydrogen or 3 hydrogen bonds. And we know these 4 bases, we named them A, T, C, and G, okay? A long time ago, people figured out that every 3 of those are interpreted together to correspond to a specific amino acid or to a specific interaction. And through years of trial and error with a few model organisms, we now know exactly what combination of 3 correspond to which amino acid every time. And we know exactly that just a very small subset of these is actually used to start new translation. So that's a site where the middleman, the ribosome, sits and starts to translate and make a machine. We know it exactly, so we can predict it computationally. When we have a gene, we can scan, when we have a long list of genes, we can scan the entire place and say, okay, this is a starting letter, this is a starting letter, this is a starting letter. And then we can find stop position by the same way. Now, this is a prediction. So to confirm it, you will need to do some complex biochemistry on the organism and demonstrate that the actual machine, the protein you find inside the organism, is exactly long as the piece you were thinking, and they correspond 1-to-1. There are exceptions, there are surprises, there are complex mechanisms, but overall, we are very good at predicting it. So for example, in bacteria, is always the same starting sequence of 3 letters for all bacteria. And so when we find those 3, I think it's ATG. Every time you find an ATG, we code for an amino acid called methionine. That's the start of a new gene, always.
When you find the start, then you find the end, you know, for.
You'll find an end somewhere.
Yeah, so the entire thing is one.
Do you want to see a real sequence data just to see what it looks like? Okay. This one, So we decided to represent the chemical variation of these bases with letters. Okay? And the way we actually write it down is literally like if it was a long text. We use actual text file to write these things down. Okay. So let's see if the computer is helping me out. So DNA chemical bases, we read only one strand of the 2. So we read only one side because we know that they pair 2 by 2. If you know one side, you can predict the other, but you don't have it. So we're reading one side. We read one side, and we represent every one of these chemical bases, we call them nucleobases, that represent this bonding. So this cross bonding here, we represent it by a single letter. And so for us, DNA data is actually mainly text files. And they're all a single long text file for most cases. And you can see there, CT, G, 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 CT, AT, T, AT, and so on. Now, there are specific programs that have been created to scan this very long text file and find specific word matching patterns. And so when you find a specific matching pattern, you can flag it as being a likely start of a gene. And then you can flag any one of these special 3 bases that are stops because they don't code for anything. So when the translating machine gets there, there's no piece matching that letter, so it's going to stop adding new pieces to the protein, adding new amino acid to its long chain. Position doesn't mean anything, I'm going to stop that. So you get that chain as long as it is. And theoretically, that's a gene. Now, it gets a little bit more complex because if it's too short, we know it's likely not a gene. If it's not similar to any other, we call it putative genes. Might be a genes, we never seen it before, we are not sure. So there are caveats beyond the prediction, but the prediction is pretty accurate in general. And in biology, all genomes, every sequence you obtain from anything is usually deposited into a large database, which is a public owned database. Biology has realized that unless you share knowledge, you're never gonna be able to interpret anything because gene interpretation is based on comparison. You compare a sequence you have to something you already know. It's like murder suspect. Yeah, you have the DNA of somebody, but unless it matches something on the database, that's an unknown person. I'm never gonna be able to find it, right? This is the same. And since 1992, in biology, every time you obtain a piece of gene of any type anywhere, you not only can compare it to a public database, but if you want to publish, you need to deposit and make it available to anyone. So it's complete openly sharing any sequence you obtain. And you can go to this one, which is, by the way, there are 3 main repositories and they're synced together. Europe, US, and Japan. Sync completely together. You put something in one, it's going to be copied in all other 3. So they're redundant in that respect. You can come here, you can put maybe with one else. Let's see. Internet helps us. Very slow this morning. If more people misspell the name of their organism. Now we're more likely. Can I get the full genome of this guy? So here you get information on the genome. This is actually the length, how many bases, 4,000,000.6 bases, ATG, so it's exactly that. You can go inside here, and you're looking at the complete genome of an organism. Here we go. Now, this is before interpreting what genes are, but people already did the entire job. So they run this through pipelines to identify the start of different things, and you can do this, and you actually visualize the genome and where the genes start and stop and what they are and so on. So each one of these lines is a gene, some of them have a name. I'm gonna zoom in. Let's do it from there. Continuous length, and for each one of these, you see has been interpreted as a gene. Let's go even more in. And you can click on a gene. And you can know exactly what this gene here is actually doing. Histidine kinase, how long it is, how many amino acids it codes for, and you can get a lot of information. At the end of the day, when we do this, we are working mainly with words and text for the large majority. Yes.
You mentioned that a certain word codes a certain amino acid, for example. Is it like always the same word always codes the same amino acid, like across organisms, or every organism has its own?
So it's pretty universal. There are changes. So far, we identified 11 translation tables different. So the general for bacteria and archaea is very well conserved, but it doesn't work as well for eukaryotes, humans, and animals, because there are a few amino acids that have a few differences. But in general, it's pretty well conserved. So many things are coded by the same 3 letters across all known life on this planet. There are things like this, together with the structure of DNA, is the same for every living being. The machinery that translates the DNA to RNA and the DNA and the RNA to proteins that makes the machines is not exactly the same, but it's very, very similar across bacteria, archaea, eukaryotes, all animal, all plants. And things like this give us clue into the fact that we have a common ancestor very early on. So that current life descends from a single type of life. Now, what we don't know, and now we're getting into more other discussion about origin of life, if there were alternative form of life early on, and our life is the only one that survived past the bottleneck of this was the only life that ever existed on the planet. But for sure, we are all connected by common ancestry. And some of the relationships are well clear. Like, for example, you are breathing today thanks to only a single event in which a bacterium got incorporated into one of your cells. And we call it today mitochondria. And that's your burning machine inside your whole cell. And you can breathe today thanks to him. Otherwise, we will not have any eukaryotes, any plants, any animal, any elephant, any yeast, nothing. So the single event of fusion between one bacteria and something that we're not sure about gave rise to all diversity today. And you have bacterial DNA in every one of your cells every day. So things like that give us a clue that we're all connected. Other questions? I realize when we speak about a few things, there are a lot of things you should know before that in order to interpret it. So ask question randomly, because if I start from the if I start from the two basics, we're never gonna get to what we need to do. So on some things, trust me, things will connect back later. On other things, you're gonna need to read or to ask questions to connect things back, okay? It's impossible to pack a 5-year degrees into a 3-day venture. But let's talk about metabolism, okay? Which is really where things connect with the geochemistry, chemistry, and geology. So life on this planet uses only two forms of energy, which is light, like plants, they use energy from photons, or chemical energy from chemical bonds and chemical reaction. Now, we know there are other forms of energy, like thermal energy, like heat. We don't know any life that can exploit a thermal difference between two reservoirs. But we know life that can exploit, for example, a chemical gradient between two reservoirs, or a redox gradient between two reservoirs. We know there are also energy in the form of radioactive X-ray or other type of radioactive particles. We don't know any life that can exploit that. So at the moment, life on this planet uses only two forms of energy: light, photons, within a certain spectrum, so within a certain energy spectrum, and chemical energy. And chemical energy is usually in the form of redox disequilibrium. So redox, we go back to redox in a second. So all life on this planet, only these two forms of energy, right? And it's very interesting to think that photon energy, plants that receive photons that can use light, they actually convert that into redox as well. So at the end of the day, all life on this planet uses redox chemistry, nothing else. Now, life does two different types of metabolism. We call it catabolism and anabolism. Literally speaking, catabolism is breaking things down to get energy. Anabolism is to use energy to build things. Like as a human, you eat food, which is complex chemicals, proteins, carbohydrates, lipids. Your body is going to break this down to get energy, and then it's going to use some pieces of what you eat together with the energy obtained to build things that are different. Like you eat a protein from the muscle of a pig or a chicken, you're not going to use that same protein to build your body. You're going to break it down into amino acid and then build up human proteins to build up your muscles. So catabolism, you get something, you break it down into products. The energy obtained goes to anabolism, which is small things that are built up to make things in your body. So your entire body is breaking down things to get energy, and then is using the energy to build new things. That's in the essence what metabolism does. And this is used to repair the cell, duplicate the cell, move the cell, to all the operation that any cell does and any organism at the end of the day. So as I said, catabolism is a set of metabolic pathway that breaks down molecule into smaller subunits, releasing energy to be used in anabolism. Anabolism uses the small building block and the energy obtained from before to build up molecules. Now, there are organisms that do a lot more anabolism than catabolism. We call these primary producers, plants. They use inorganics from atmosphere, like N2 and CO2, and from the ground, like phosphate, nitrate, inorganic chemicals, together with the energy that they're able to derive from photons and redox chemistry to build sugars, to build the entire plant. And then the plant is eaten by animals. And in this case, animals, they're dominated by catabolism. They also do anabolism. We also build muscle in our body and cells and everything. But in order to do that, we cannot start from inorganic chemicals. We need to take somebody, chemical, break them down and rebuild. That will be what the basic difference between autotrophy or autotrophs, primary producers, plants, but not only, as we'll see later in microbes, and heterotrophs, secondary consumers. They're also called when you were in high school. All the animal, herbivores, carnivores, all the other things around. This slide here is probably the most important concept in metabolism. If you're able to get this one in your brain, you're able to ask the right question all the time. Any living being on this planet needs a carbon source. You need carbon to build your body because carbon, we are based on carbon chemistry. Lipids are carbon, carbohydrates are carbon, proteins contain carbon, everything in our body contains organic carbon. So somewhere you need to pick up carbon to grow and build things and duplicate. So every living being on this planet needs a carbon source. And then you need an electron donor and an electron acceptor, an energy source and a dump at the end. Now, we'll make a lot of examples for this, but remember that any metabolism you can imagine out there needs electron donor, carbon acceptor. The energy from the source is used to metabolize the carbon and then is dumped somewhere. Now, it'll be more clear in a second, but remember, every life form out there needs an electron donor, also called energy source, where the energy is coming from, where the electrons literally are coming from, a carbon source, and what we call electron acceptor. The electron acceptor for you today is oxygen. That's where you're dumping your electron into while you're respiring. We'll get to that in a second. But this one, we repeat this a few times, okay? So we said two forms of energy, lights and chemistry, but both of them, they boil down usually to redox chemistry, redox disequilibria. And you remember redox from your school, right? Reduction, oxidation, reactions are type of chemical reaction involved a transfer of electron between two species because oxidation states are changing, right? One is going from reduced to oxidized, one is going to oxidize to reduce. Okay, that's this change in redox state, in redox chemistry. And life is designed exactly to do only this. Life does this. When you think of proteins, especially specialized proteins, you need to think of proteins like well-designed three-dimensional structures that can interact with things. In the case of redox chemistry, these are very complex structures that you should see just as a good way to keep some tiny metal in line. These are the metal center on this big protein here for electron to move. So in proteins, in redox proteins, which we call oxidoreductases, because they do redox chemistry, what you have is a series of metal centers where electrons are moving to. And we'll see what they move from to in a second. But complex protein, you should see them simply as a transducer of electricity. They're literally passing a current through, okay? Electrons are moving through it. And in essence, what we do as a cell, all cells on this planet, you get, let's do the opposite, let's start from here. You know that in redox chemistry, you need two reactions happening at the same time. We just said one is reducing and one is oxidizing, right? So the chemistry that is oxidizing is giving away electron. It's moving from reduced to oxidized. This electron jump to the next acceptor, the acceptor, the terminal one, which is getting reduced. It's the same way a battery works, like the Volta battery. I don't know if you remember that as an innovation. Volta realized that there are metals, when you put them side by side, they have a galvanic current. One becomes a donor on electron, one becomes an acceptor. And you get an electron flow between one to the other. And one of the metal is oxidizing and the other one is reducing. And Volta realized that if you put things attaching to each other, you have electron and proton exchange quickly. But if you put a space between them of water with ions where protons can travel, but electrons cannot, and you connect the two metals with a wire, you are breaking this reaction down. Proton go one way, electron go the other way, and you can use the electron running into the wire to make work. That's how our electricity system works, basically, continuous current at least. So you get two chemicals that really want to react with each other, but you make the reaction go to a forced path. And that forced path allows you to harness this energy between the chemistry to do work. Well, that's how a battery works, Volta battery. You got the protons moving from one to the other, and you got the electrons going the other way, meeting at the end. Cells work exactly like this. We have a redox reaction of a donor and acceptor that do not meet together. They're separated in space. protons are going one way and electrons are going another way. And because you split these two, you can actually do work. We literally work as a battery. Now think about two very good electron donor and acceptor. Hydrogen, powerful reductant, right? Tendency to give electron very quickly to a lot of other things, and oxygen, which is a powerful oxidant. And we know because it's everywhere, you rust metal, you oxidize and break down things. Why? Because it's a powerful acceptor of electron. It's an oxidant because it can strip electron very quickly. And you know that hydrogen and oxygen react very well together. It's pretty dangerous to keep them together. We actually use them as propeller for rockets, or we have been using it as propeller for rockets for a long time. So oxygen and hydrogen, when they react together, they can free an enormous amount of energy, right? What happens within the cell, and there are cells, they use hydrogen and oxygen together, but they don't let them react together. Hydrogen gives the electron on one side, the electron is passed around, and we get to that in a second, and then it finally gets to oxygen. So the net reaction is the same, like when you burn oxygen and hydrogen together, but it's split in very small amount of energy freed each time. It's not a combustion, otherwise cell would explode, right? So in principle, we work like a battery. We get one reaction, protons go one way, and then electrons keep flowing within an electrical system, which is made by different proteins that contain metals where electron can just jump. And actually it's interesting for physicists, engineers, if you measure the distance of these metals within a cell, within a protein, it's always about 14 angstrom on average, which is the best gap to have electron tunneling, so the electron to jump from one point to the other. It's very well calibrated, the system.
That's the gap between the two centers.
Yes, two metal center within a protein on average is about 14 angstrom, which is optimal distance for electron tunneling, for electron jumping from one place to the other. And then the protein, the one protein that has everything on it, will move close to another protein, the electron will jump to the next, jump inside, they'll get close, jump to the other, jump inside, and they keep jumping around. Why life needs to separate the two alpha reaction, this chemical reaction, and things jump around? Well, to use energy to do something, right? We get to that in a second again. So remember, every time we have a redox chemistry, we have a donor of electron, and then we have an acceptor, and things are coupled together. Works for batteries, works for life, all the time. And what we do usually in biology, or well, this comes actually from inorganic chemistry, but what we do, we can align different oxidation redox couple along a scale that goes from negative to positive. We call it redox tower, and we can calculate how much energy is in that redox, right? The millivolt differential that are there. And you can see at the top, for example, we have sulfate to sulfite, CO2 to glucose, two protons to hydrogen, CO2 to methanol, and you can think about changes here. For example, nitrate to nitrite. That's a specific jump, gives away two electrons, is a reduction, and has a certain point potential, a certain tendency to happen and give electron away. And what you can do, you can get any two of these two, couple them together, like down there, and calculate the actual amount of joules of energy that that reaction is giving away. For example, hydrogen plus nitrate, so a good donor like hydrogen, plus nitrate, a good acceptor, will result in making nitrite plus water, and as a negative delta G, I don't know if you remember the Gibbs free energy. When it's negative, you have a spontaneous reaction, provides energy. When it's positive, you need to provide energy. Well, redox reaction in biology, they're usually negative because the cell needs to get energy out of it. Cell also do positive delta G reaction, but in that case, they need to put in energy to make it happen. We are looking for an energy conserving reaction that give energy to the cell to do other things. So we do this and we can do calculation and we can say exactly how much energy is in that redox reaction. Now, this is very misleading because it makes you think that the reaction is happening like in a bottle, right? We put hydrogen plus sulfate. This is relevant for underground hydrogen storage. hydrogen plus sulfate in acidic media gives you HS plus H2S with a good negative delta G. So what you are getting there is what we call sulfate reduction, and we know can produce sulfide, which is very bad for the reservoir, like that's the gas, the ion species we don't want. The reaction is misleading because can happen also abiotically, can happen in a pot, needs to warm up a lot. But if you put sulfate and hydrogen and you warm it up, you're gonna get H2S formation. But why we write it the same for life, it doesn't happen together. It's separate. So hydrogen oxidation is happening somewhere, sulfate reduction is happening somewhere else in the cell, and electrons are traveling around for the cell to do work. Why they're traveling around? Imagine delta G gives free energy to be the total amount of energy that a reaction can give you, okay? Imagine it like potential energy. We have water on a hill, and then we can make it fall down to the ground, and the total height is going to be mgh, right? Potential energy. Now, if that jump happens in a single jump, there's not much you can do with the water. It's overpowering. There's an entire waterfall coming down instantly. Can be dangerous. So that would be your chemical reaction happening in one pot, right? Boom, things react. Hydrogen, oxygen, flame, boom, they explode. But if you can split this and have the total potential energy, not to jump in a single jump, but to do smaller jumps. At each point, we can intercept that jump in the world. That's what life does. You have a donor, an acceptor like oxygen. That's happening in your body right now while we're speaking. And then that donor there is not giving the electron directly to oxygen, which will be thermodynamically too powerful and wasting a lot of energy in heat, because we know when reactions are not controlled, heat is produced, but it's passing it to a second acceptor, which is a weak acceptor. It's like me throwing something to you back there, chances we break things and we do things wrong a lot, or we pass it around all the way to the back. And at each step, somebody can do something with what it gets. So cell do this. You break it down, and you have 1, 2, 3, 4, 5, X number of carriers on the way. So the total reaction is the same, but you are splitting it in smaller reactions that are happening along the way. And you see it here. From the first one, it goes to the second, to the third, to the fourth, and so on until it gets to the last one, which is oxygen getting the electron at the end. And each one of these steps has the exact position in the getting electron and giving electron, which is able to get electron from you because it's less powerful than the one before, and then it's more powerful than the other is giving it down. So there's a hierarchy on where electrons go, okay? It's like having a circuit where tension is falling through the circuit and you're passing things slowly from one to the other. Literally works the same. And the electron and the donor, in many cases, they can be inorganic or organic chemicals. Life doesn't care, okay? For example, in your body right now, the donor, the first donor of things is glucose, which is a reduced carbon. So the sugar you just had from the coffee and the croissant outside is going to provide an electron for a reaction that contains at the other side the oxygen. You are breathing oxygen, which means you are dumping an electron into the oxygen. Now, the sugar happens to be also your carbon source. So you remember I said you need an electron donor, an electron acceptor to do redox chemistry, and then you can use those electrons to do something, and you use the carbon for that something. In this case, your electron donor is glucose, your electron acceptor is oxygen, your carbon source is glucose. And you can identify these three for every living being out there. Chemicals can be sometimes donor, sometimes acceptor, sometimes both. How? Well, you know redox, oxidation states of a chemical, right? Sulfur, for example. How many oxidation states has sulfur? The sulfur atom. Basic chemistry.
-2 to 4.
-2 to +4. That's the entire range, right? That it can go. Now, naturally, if you are at the -2 state, can you be reduced more? No. So you cannot accept more electron. You are in your lowest state. So the guys that are at -2 can only be electron donor. Can only give away. +4, sorry, +6, not +4, sulfate, +6 cannot give away more electron, has given away all the electron of his outer shell. There's nothing to give away, can only receive. So it's only an acceptor. So basically, every time you see a chemical, and you know the total oxidation state that can go, if you are at either end of that scale, you can only either give or receive. If you are in the middle of that, well, you can do both. It's going to depend who your partner is, right? So sulfide to an extent, only electron acceptor. Everything in the middle can be both electron acceptor and donor. And then once you get to -2, you're only a donor. There's not much you can do. Carbon is the same. What are the oxidation states of carbon? +4, and what's the other end of the scale? What? 0 would be graphite, but we know carbon can become also more reduced than graphite. For example, methane. Methane is one of the most reduced carbon. What's the oxidation state of carbon in methane? CH4. -4. So we go from +4 to -4, right? So if you are methane, you can only give away electron. You are in this reduced state. If you have CO2, you can only get electron. If you are in the middle, you can do both. So it's very easy to identify donor, theoretical donor, and theoretical acceptor. Because if you are in a very reduced state, you are a donor. If you are in a very oxidized state, you're an acceptor. And in between depends who you're dealing with, and we'll get to that. By the way, sulfate and hydrogen sulfide are the two ends of the sulfur scale, all right? And they're of interest in underground hydrogen storage because we don't want sulfide. And everybody's thinking about sulfate reduction happening in a reservoir. But every other chemical element of sulfur in this slide here can be used by microbes. So if you don't have sulfate in your reservoir, that's good news. But you might have elemental sulfur, or thiosulfate, or polysulfide, or organic sulfur, or pyrite. They can still be reduced all the way to H2S. So looking only at the two ends of the scale is very limiting. But that's what's happening in underground hydrogen storage right now. You know, the things are more complex than that because microbes can use all of these as donor or acceptor. And if they use as acceptor, they're going to be reduced. They're going to become H2S or HS minus. And this is an example. These are oxidation state of common things. We have hydrogen, carbon, oxygen, nitrogen, sulfur, iron, manganese, arsenic, selenium. All of them can be used by microbes and many more. And you can see hydrogen up there is either 0 plus 1. There's no other way. So it can only give electron. It's not a very good acceptor. And then you got carbon, -4 to +4. Oxygen. Oxygen is very good only at getting electron and becoming water. The other way around is very complicated. Only plant can do it. Only plant can strip an electron from water. And they do it thanks to photon. They use the energy from the photon to split water and get an electron and they free oxygen. That's how plants make oxygen. But this reaction in this way is very easy. We all do it. This way is very hard, very complicated. You need to provide a lot of energy. And they provide energy from the sun in that case. And then you can see, look at nitrogen, from ammonia to nitrate. Look how many oxidation states. -3 to +6. Sulfur, iron. We have iron 0, iron 2, iron 3. Microbes are very, very good at playing with iron 2 and iron 3. So iron 2, which is reduced, is going to be the donor of an electron. Iron 3, which is the oxidized form of iron, can actually receive an electron, can be a terminal electron acceptor. The same goes for arsenic, for example. Arsenate to arsenate. One can be a donor, one can be an acceptor. Selenium, tellurium, uranium, ytterbium. We can do a very long list of things that microbe can either get electron from or give electron to, okay? Any questions so far? Probably a million. No?
No.
So every time you see something, every time we see a microbial metabolism, we look at a few in a second, you can always identify, in the case of underground hydrogen storage, for example, hydrogen and sulfate. This is the electron donor, this is the electron acceptor, and we can calculate how much energy is there. Now, typically, the Gibbs free energy of the reaction, negative tells you that the reaction is spontaneous. The bigger the number is, it tells you that there is more energy going on there, like minus 200 gives you more energy than minus 50, right? The problem is that we calculate this usually at standard condition. Do you know what laboratory standard condition are? Have you ever heard the term standard condition? What does it mean? Temperature, pressure, pH, ionic strength, and concentration. So we're considering hydrogen and sulfate to be 1 molar each. We are considering pH to be 7, atmospheric pressure 1 bar, 25 degrees, 0 concentration of the reductants. But we know that rarely we're going to ever see this in natural environment. First, 1 molar is very unlikely to find around, probably different concentration. If you are in water or not, speciation of things might change. Usually the ionic strength of your media is not 0, but there are other ions going through it. You rarely at pH 7. In underground hydrogen, in underground reservoirs, you never at 25 degrees, probably higher, and you never at 1 bar pressure. Even if you just consider the hydrostatic pressure above the place where you're working, it's probably 50 to 150, 200 to 300 bar. That changes the actual energy of the reaction. In this way, this is an example of a reaction. At the top, this is H2S plus water equals sulfate plus HS. Now, if you calculate the Gibbs free energy at different pH and different concentration of HS present in the media, you see that the energy can vary a lot. Like if you are up here, there's 0 energy to be gained. If you are down here, there can be up to a lot of energy to be gained. If you are in alkaline condition with very low concentration of HS minus, which is one of the products there's no accumulation of the product, you have a lot of energy. So one of the challenges right now for underground hydrogen storage is to be able to interpret not just what metabolism is possible in theory, but what metabolism is possible at in situ condition in your specific reservoir. And as you see, the delta G goes from 0 to minus 120. That goes from a reaction that will never happen to a reaction that gives away a lot of energy from microbes. And it's mainly due to the change in pH from 6 to 8.5. That's not a huge range. There's a couple of pH units. And getting your product very low. So if that HS minus at the product side of the reaction is then reacting with iron, for example, and precipitating as pyrite, well, you keep removing the products, means there's no stop at the reaction, can keep going, can provide a lot of energy. So in situ actual energy of a reaction is very different than the standard laboratory condition. Take any paper today about microbiology and underground hydrogen storage, they always calculate energy as standard in situ condition. Doesn't really tell you anything about what's going on in your reservoir. Let's talk about this reaction though, because I told you that, for example, hydrogen and oxygen are a very powerful electron donor. Like hydrogen is very good at giving electron. Oxygen is a very powerful oxidant. And we know that if we put these two together, we can actually have an explosion. The thermal energy freed by this reaction is huge. And yet you know that, for example, when you are in your kitchen and you switch on your burner, you provide methane and there's oxygen around, but it doesn't start fire immediately. You need to give it a little kick, right? You need a spark, you need something. Why? Because many of these reactions have an activation energy. The reaction doesn't start unless you provide a small kick to get it going. Now, this is commonly represented. You have progression of the reaction, free energy. Before the reaction can go down and give you that potential energy, delta G, you need to overcome a small barrier. And that barrier needs you to provide a little energy. That's what you do in your kitchen. Before you burn methane with oxygen, you need to provide with the flame a little bit of energy so that things get going. Now, biology is very, very good at reducing that activation energy. So biology can make things happen even if when the reaction has a block, is locked. Or biology is very, very good at making happening fast things that are very, very slow. Now, let's talk about this because I think it's one of the most important concepts when you model anything in real life out there. Think about the oxidation of iron up there. We know iron 2 is reduced iron and reacts very, very well with oxygen, right? If you put iron, reduced iron outside into the open atmosphere, it's going to start to rust. The rusting is iron 2 becoming iron 3 and then getting hydrolyzed become iron hydroxide, FeOH3. And you make it rust essentially. And happens all the time. Every time you put some reduced iron outside, it's going to start to oxidize because there is oxygen. The reaction is spontaneous. If you calculate the Gibbs energy of the reaction is negative, there's a good amount of energy to be gained from oxidizing iron with oxygen. That's a spontaneous reaction, but it's also a reaction that many microbes do. Many microbes will get iron 2, will get oxygen, and will strip an electron from iron 2, will become iron 3. They will use it for stuff, and then we'll dump it on oxygen, which will become water, and then cross hydrolyze with iron 3. So if we look at this reaction, iron 2 plus oxygen in aqueous media will give you iron 3 rust, iron hydroxide that will precipitate. And we know the reaction is starting, reactions are bidirectional usually, right? You start to go in one direction until you accumulate too much product and it doesn't really flow anymore, it stops. So you see reaction goes forward and reaches the equilibrium. Now, for biology to use this reaction, you need the reaction to be slow, very slow. It's like, let's go back to the waterfall analogy of before. So you have a waterfall coming down, and you want to start to build intermediate passage to be able to use the water. But if the water is coming down already too violently, there's no way you're going to be able to do any work there, right? In engineering, we would need to divert the river somewhere else, do all the work, and then bring it back. Well, when chemistry is already happening on its own very fast, biology cannot really deal with it. If oxygen and hydrogen are already making a flame, there's no way for biology to control the reaction. Biology needs the reaction either to be locked because of activation energy or being very slow. And we see this very, very well. If we go in springs where there's a lot of iron 2 coming from underground and there's oxygen from the atmosphere, and we start to measure this reaction, right? We measure the reaction. This K here is the constant, the speed at which the reaction is happening. And we do it in many springs. And we try to do it in springs where there are live microbes, and then we kill the microbes. And we measure also the spontaneous reaction, the abiotic reaction. You can see here, live microbes in red, dead controls in black. pH is on the x-axis. So when you are acidic, biology is faster than geology. This speed, right? Minus log. So higher, faster. Faster, 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 faster. They get pretty close to each other. Now, biology is similar in speed than geology until you don't see any more red dot on the second half of that. That's because the spontaneous geological reaction becomes so fast at neutral pH to alkaline pH. The biology is not touching it anymore. So the delta G of the reaction is exactly the same. Thermodynamics tells you that the reaction is spontaneous and tells you how much energy you have, but it tells you nothing about the rates. It doesn't tell you nothing about who's making it. In iron, if you calculate the percentage of how much biology to geology is making the reaction, you get something like this. A low pH, 90% or more of the reaction is biological. 90% or more of the total reaction of oxidation of iron is biological. In those pools, biology is the one oxidizing iron, the majority of it. More than 9 atoms out of 10 in every minute or second or unit of time. As pH gets more neutral, biology contribution drops, goes around 50, and then biology gives up completely. If you go to a pool that is pH 80, there's still iron oxidation going on. Probably faster than the low pH as total reaction. That's purely abiotic. Life is not touching the reaction anymore. Why? It's too fast. So one of my mentors used to say, things that burst into flame are not good to eat. sugar is reduced carbon. You know you can burn sugar. Have you ever tried? Like if you get a packet of dust sugar, powder sugar, how you call it, the one we put on top of cakes, and you suspend it in air and you flame it, it's going to actually explode. I don't know if you ever heard that meals that produce flour are highly explosive environment. Because if you mix flour, which is essentially sugars, into air with a certain ratio, just a spark is going to make the entire place explode. Go on Google and search for it, because they're very, very dangerous environment. So sugar is a very, very good fuel source, and you can burn it very, very well. But if you leave it on your countertop, there is oxygen around, sugar is there, nothing is happening. Why? Because the reaction, unless you heat it up a lot and you provide a lot of starting energy, your sugar is oxidizing, but it's so slow that it's going to take tens to hundreds of years to oxidize. And yet biology is very good at using sugar and oxygen to make energy constantly in your body. Why? Because biology is very good at speeding up slow reaction or removing activation barriers. So in essence, you need to worry about biology when you have a redox reaction in your system, and you know the abiotic reaction is either stuck because of activation energy, requires a catalyzer, or is very, very slow at happening. Then biology can overcome this barrier and be the maximum contributor to your reaction. Think about iron oxidation. Depending on pH, biology can be responsible for all iron oxidation or none. That's a pretty huge change, right? And it's dominated by pH in this case. Unfortunately, for many reactions of interest, we don't know the relative contribution of biology to geology. We know they're almost always overlapping when you are within the domain of life. We said yesterday the domain of life, less than 135 degrees, any pH, any temperature, any pressure, any salinity up to saturation, and enough pore space for things to survive. That would be the domain of life. So if you are there, chances are that your abiotic rate and biological rate are overlapping, and you need to figure out the relative contribution. Because dealing with an abiotic reaction is one problem. Dealing with a biological reaction is a different set of problems. In other words, this is the same things in a different diagrams. We have the log of the reaction rate from slow to fast. We have the biological contribution, dashed line, and then we have changing environmental condition. So at any point in time, you have abiotic reaction and biological reaction contributing together, right? And the relative contribution is to be defined. In this case, I'm showing it as very high biological contribution to start. As conditions change, the two rates will modify. More often than not, the abiotic rate is the one changing a lot. It's becoming faster and faster and faster until it becomes so fast, the biology drops to zero and only abiotic rates are present. Now, if you can figure out for a reaction you want or you don't want to happen in your reservoir, where the trade-off is and what are the controls, then you can move around. You can say, hey, I don't want hydrogen to react with CO2 because I don't want methane to be made, but I don't want hydrogen to be used with sulfate. So I'm going to go in a place where sulfate reduction is completely inhibited biologically, because we are on the second part of my graph, and biology is now contributing to that. And I know the abiotic rate of sulfate reduction at this temperature is so slow that I can have that reservoir full with hydrogen for 100 years before any H2S will start to accumulate. So the real big challenge right now for the future is be able to figure out where these trade-offs are, where they're not, and how to control them. I think that this line here, with the understanding that if you are, the caveat is if you are in the domain of life, right? If you are below 135 within the condition we set, understanding what's the relative contribution of biological and geological reaction for any chemical reaction you might happen in your reservoir is our biggest challenge. That's where the money is right now. And that's where a lot of the work we're doing goes in that direction. And I think this one here is probably the most important slide of all today. So to now, when we take a small break, remember that Life uses redox energy. So if there is a redox couple that might react, life can theoretically use it. And life is going to use it only if the spontaneous reaction, because to get energy needs to be spontaneous, right? You need negative delta G. Is either slow, and life can make it faster, or there's an activation energy. So any redox reaction that you can imagine in your reservoir, in your system, probably there's an overlap of biological to abiotic reaction happening at the same time, like everywhere on the planet right now. And figuring out where you are, that's where the real problem of today is. Before we get a break, questions so far?
Yes, one question.
Go.
Very interesting. That diagram, I'm not to summarize my misunderstanding of biology so far. My question is that one, are you going to speak later about the reservoir condition? When
Yes.
you are basically transferring that, basically.
Yes, tomorrow. Tomorrow
Because
morning.
my question is, what can be in a reservoir the activation process or the activation energy, where it can come from? In my understanding from a lot of situation experiments shown is that sometimes some experiments show that nothing happened. Similar experiment done in a different context, probably with a different chemical environment, I understand. Some of those reactions happen very fast.
You know,
So is it what part of what you call the heterogeneity complexity
Also.
of the reservoir? So are you suggesting that each reservoir needs to be studied from scratch to understand which are the boundary condition of chemistry, then?
So we speak more about this tomorrow when we talk about
Okay.
reservoir, but we need first to figure out, so unless we want to treat every reservoir as a new case study, we need to figure out first principle that govern this variability, right? In order to do this, in order for a reaction to happen, let's pick up a reaction that is abiotically is very, very slow, right? And potentially like this one, potentially biology, and let's say our reservoir condition are here. Reaction is very, very slow. This is log, abiotically, but biology has the potential to make it very, very fast. The potential why? Because you also need the right biology to be there to start with. So if the reaction is there, does not mean that somebody is actually there to do it. It might be that in that reservoir, there's no one to do it. There's one additional caveat. We said that to control redox chemistry, proteins have inside specific metals that can pass electrons around. Those metals are a long list of different metals. Sometimes you need nickel, copper, cobalt, molybdenum, vanadium, tungsten. Sometimes you might be there, but you don't have enough nickel to build your proteins because you are in depleted nickel condition. That's actually the subject of work of my ERC grant, how to tinker with trace elements to modulate activity. Third, in the subsurface, when you look at chemical reaction, like this one, there's a lot of iron too in a lot of rocks in the subsurface. What is missing? Well, most of the case, you're limited in oxygen. You are limited in good oxidants in subsurface reservoir. So unless you are providing by operation oxidants by mistakes, a lot of the very powerful reactions are inhibited because you're missing the electron acceptor. And the only electron acceptor you have in the subsurface are weak electron acceptor, iron 3, CO2. If you're unlucky, you are in marine conditions, sulfate, because you are with seawater entrainment around. Otherwise, all that sulfate is virtually absent unless you drill through gypsum layer or stuff like that. So you're breaking sulfate into your reservoir. But this is exactly the type of thinking we need. You know, theoretically speaking, there are people that get brine from reservoir, and then they say potential for a sulfate reduction. They give 1 molar sulfate, a lot of organic carbon, and of course somebody's going to use it at that point. But if you do it in situ condition, nobody's doing the reaction because they're limited in sulfate or they're limited in sugars and carbon source. Remember the triangle there. You need electron donor, carbon source, and electron acceptor. Subsurface is generally limited in carbon and acceptor, plenty of donor. Surface environment are generally limited in electron donor, plenty of acceptors. And so you need to balance all three. And I think there's a lot of things we don't understand right now of first principle controlling this. So right now we need to approach each reservoir as an individual experiment. If we can get to general principle, it's going to be easier. because, you know, chemical composition of the reservoir, in situ pressure, temperature, pH condition, they can be known relatively easily. So we can potentially construct models of how things are going there quickly. And tomorrow we will discuss some of these models, some of the efforts that people like Martina and Guillermo and other people in my lab, Gabriella, they've been doing to try to get to some of the principles that control this.
So are you suggesting that, okay, in the real context of the subsurface wells, what we got wells, the wells which are injecting stuff. When you injecting stuff, let's say you are injecting hydrogen, you are injecting a bit of oxygen inside. It's impossible to inject. So those are essentially the perturbation of your context that you have to take into account.
Like specifying how much oxygen becomes dangerous for the system. Specifying that whatever emulsifier you're using is also an electron donor and a carbon source to the community. We know this from petroil bioremediation. Dispersant have been used for a long time at sea. You have an oil patch created, you go up with plane and you drop dispersants. Dispersants work like soap. They have an aliphatic and an aliphilic chain. They put into oil and they disperse it in water. Well, they're made of carbon, nitrogen, oxygen. They're basically very, very good substance for microbes. And people showed that if you use dispersant, the microbes that normally would eat the oil, they start to eat the dispersant. So you moving them away from breaking down the oil, you're making them break the dispersant. And you don't see the petroleum anymore, but now your community is feeding on your dispersant rather than on the oil itself. So all these caveats will need to be worked out in operations. And at the end of the day, having first principle that suggests what are the critical parameters will provide engineers with boundary constraint on where to find solutions to problems, right? Yes.
I don't have a background, but does these reactions usually happen in the gas phase or liquid phase, or doesn't it matter at all?
Well, microbes tend to live in liquid or attached to surfaces. Now, there are microbes that in order to use a chemical, they need the ionic form of the dissolved form. And there are other microbes that use very, very well gases directly from the atmosphere. So we have it both ways, right? I would say that if you are in a pure gas phase, microbes cannot really do much. But in porous reservoir, you are not in pure gas phase because you have a lot of surface area, right? So microbes will be attached there. Plus you have wettability of the system. So you have a thin layer of liquids attached around, and microbes will thrive there very, very well. And the big question right now is in salt caverns, because in salt caverns, you have a very small surface area compared to the volume where you're storing the hydrogen. So with respecting this place, microbe will play a very, very minor role in problems in salt caverns. One is going to be huge in aquifers and porous reservoirs. But this is just an extrapolation of what we know from other things. We haven't checked yet. We've been trying to extract DNA from salt cores right now, but it's a bit challenging. There are microbes discovered in the last 10 years that can use, so we always knew the microbe can use hydrogen as an electron donor, can use methane as an electron donor, a reducer carbon, but also carbon monoxide. Because carbon monoxide is +2. You can push it to dioxide, which is +4. So you can still get a couple of electrons out. And we always knew there are microbes that use all three, okay? Hydrogen, methane, and carbon monoxide. Now, we always thought that you need a good amount of it to be usable. So you need certain good concentration. So we always thought that things they use methane needs to be methane seeps and well. Things they use hydrogen needs to be close to a source of hydrogen. But in the last 10 years, we have identified the soil microorganisms that can use the trace gas concentration in the atmosphere to get energy. Hydrogen in the atmosphere is 1.7 ppb part per billion. And yet there are microbes that live extracting a 1.7 part per billion atom out of the atmosphere, and they make a living out of it directly from the gas phase. So the interaction with the gas phase is very, very good. They just need to be attached somewhere.
Are those microbes just the kind of energy transmissions, like from hydrogen, just?
No, they're not obligate, but they supplement up to 30% of their metabolism using trace gases. So they do not depend only on that. but they use it very, very efficiently. Well, the problem is trade-off, right? So in biology, we have two parameters that describe how an organism, especially a single enzyme, interacts with things. One is the speed at which it can catalyze the reaction. The other one, we call it affinity. How good that enzyme is at finding the substrate. So when your substrate is very, very abundant, you don't need to be very good at finding it because it's going to just happen to be there and you can catalyze the reaction very, very quickly. When your substrate is very, very rare, you need an enzyme that can really attract the single molecule floating around to it. When that happens, the binding energy is so high that then the reaction is very, very slow. So basically, biology can either do things fast or can do things with small concentrations, but not both. So theoretically, yes, we could use these trace gas-eating microbes to extract things, but reaction will be super slow. So I won't know if it makes any sense. There's this big trade-off between specificity and affinity and speed, which I think is common to other trade-off laws that are out there, right? You can do things right, or you can do them fast, not both at the same time. Any other question, or we take a 5-minute break?
And next question, so getting back to this diagram, you got slow to fast versus change environmental condition.
Yes.
Still, environmental condition for is it pressure, temperature, fluid content?
Anything. We don't know.
So you essentially, our game is to transform this diagram into a PT fluid.
Exactly. This is a theoretical diagram, right? So if you're considering the iron oxidation reaction we were looking at before, the biggest parameter influencing the speed of that reaction is pH, by far, in iron oxidation, by far. Of course, if you keep oxygen availability the same, if you reduce the oxygen to macroaerophilic, where the abiotic reaction stays low because the interaction of iron with oxygen is very, very rare, microbes can do it for longer because they're very good at attracting. They have high affinity enzymes to attract the oxygen. So if you think about the landscape of these parameters, that's going to be a very complex multivariable landscape. You know, in some corner, pH is going to totally dominate your slope. But if you lower another parameter, for example, if you increase pressure, then you go back up on the other corner. So this is a multiverse of parameters. And we're still exploring one parameter at a time, most of the time. Because combinatorial space, you know what combinatorial space is, right? Does anybody know? No. So let's say you want to control pH, one variable. So you do your measurement against pH, one variable. Let's say you do it every 10th step across the scale. Okay, that's 10 conditions you measure. And now we're going to do it pH and temperature. And you want to measure temperature with 0 and 100 every 10 degrees. That's 10 by 10 is 100 conditions you need to test. And then we're going to add salinity. Salinity is a third variable. And they say we're going to do it in 10 steps. Now it's 10, 10, 10. And the combination of parameters explodes into your space until what we call combinatorial space becomes n per p per x per y becomes huge. Nobody's going to ever be able to test those things. That's why we need few well-designed experiments to get first principle. And then we're going to need to predict what responses are across the scale. Because doing the combinatorics of this experiment is just impossible. Things just explode in your face so fast. And then if you consider that in biology we do duplicate and triplicate, and each one of these requires time. So another variable in every experiment is how often you measure the rates. Every hour, every 2 hours, every day, for how long? 1 month, 3 months, 6 months, a year. And then suddenly you have 10,000 samples you need to run and analyze. And each one of those requires 10-hour analysis time, and you need the lifetime of your PhD students just to run a single experiment. So, yeah. 5-minute break. If you have a question from the previous part, we push them to the end, okay? So we can do some things together now. So, as I said, let's take any compound. You have reduced state, oxidized state, they can do things. For example, look at the top, nitrate, +5. That's the most oxidized form of nitrogen out there. Then you can do things like hydrogen, very good electron donor, nitrate as electron acceptor. The product, if you go all the way, it's ammonia. Look, you go to +5, -3. That reaction up there is giving you 8 electrons for every molecule you react. You need a lot of hydrogen to react with only 1 molecule of nitrate, and will give you 8 good electrons to do work. Huge energy, a lot of electrons. We call that nitrate reduction to ammonia. Pretty simple, right? So it's hydrogen oxidation linked to nitrate reduction to ammonia. And then you can do the opposite. You can go from ammonia up. So ammonia becomes your electron donor because it's the other end of the scale. It's a very good electron donor. You couple it to a good electron acceptor, in this case, oxygen. So you can do ammonia oxidation to what? Nitrite, in this case. So you go from -3 all the way to +3. That's 6 good electrons that you want to dump on oxygen and make water. You can do more creative things in biology, what we call nitrogen comproportionation. So in this case, the electron donor and the electron acceptor are both nitrogen-containing molecules. We're using ammonia as a donor, nitrite as an acceptor. We're smashing them together, and we're going from +3 to 0 and -3 to 0. That's 3 electrons shuffling around, and we're stopping in the center. So in this case, we don't have separate donor and acceptor from 2 different molecules like hydrogen or oxygen. We're doing it within the system. We do also the opposite. You start from the center and you move somewhere else. So you get 2 molecules of nitrite, and then you split it to nitrate and N2. Boom. We call this disproportionation. Happens with thiosulfate a lot, for example. Thiosulfate, which is a midway of sulfur, a portion is oxidized to sulfate, becomes sulfuric acid, and a portion is reduced to elemental sulfur. So you take the electron of reducing one and you smash them on oxidizing the other. Now, all these creative reaction going from oxidized to reduced to oxidized, midway splitting, 2 of them together, they're all carried out by biology. All possible. And we give them a special name. DNRA, ammonia oxidation, and ammox. The last one is actually unknown in biology. Theoretically valid. We don't know if microbes do it.
None of them can work without biology.
Well, all of them are negative delta G. But in nature and normal condition, they're so slow that they don't really work. The problem is that they either require a catalyst to start, or they're very, very slow.
So in this equation, you have to add, in addition, for me, the free energy gives energy and the speed. Otherwise,
yeah, you need the abiotic rate, and then hopefully biotic rates. At the moment, the problem right now is that everybody's thinking in terms of thermodynamics, which is very, very good to get if things are likely to happen alone and how much energy is there. But they're not telling you if they're happening and how fast they're happening. And modeling without rates is completely useless because we want to know how fast things are going. We don't just want to know if theoretically they go, right? So rates is the big elephant in the room at the interface between biology and everything else. Engineering, geology, biogeochemistry, chemistry, and so on. Rates. We're all after rates right now, but they're hard. Because if you manipulate your system, you alter the rates you're measuring. We'll get to that tomorrow a little more. So, yes.
One reaction can have different rates.
Oh, yes. Yes. Completely different rates. Completely valid.
And what are the rates? Yes. What is manipulating the rates? Like conditions or?
Can be organism type. Different organisms are faster or slower. Can be environmental conditions. Can be side reaction of other things that are happening. Remember yesterday, biochemical cycles are connected. Now, when you look at this organism here, look at the top. Hydrogen plus nitrogen gives you ammonia. Fantastic. The cell uses that to get energy. To do what? To grow and duplicate. But to make another cell, you don't just need hydrogen and nitrate. You need also carbon. You need also phosphorus. So maybe the theoretical reaction can happen very fast. But cells are doing it slow because they're limited in carbon or they're limited in phosphorus. The reaction up there requires two key enzymes. The first one is the one using hydrogen. We'll speak about them tomorrow. Usually they contain nickel and iron inside as catalyst to make the hydrogen oxidation go faster. The nitrate reduction for ammonia requires 6 different enzymes, of which the most important one contains molybdenum as an element to give the electron to nitrate. If there's no molybdenum around, you have the theoretical lots of energy. The rate might be higher in an optimal condition, but there's no molybdenum. Nobody can use the nitrate, that reaction doesn't happen. So there are a lot of secondary constraints on this. That's why I was telling you, in situ free energy and in situ rates are what we need today, and we don't have. Because you may have excess hydrogen and excess nitrate, but still cells are not growing because they're limited in carbon or limited in iron. In the ocean today, if you go in places like the Pacific and Antarctica, you have everything you need to make the water green in photosynthesis and tiny photosynthesizing microbes. But they don't do it, it's transparent. Why? They miss iron. Those places are iron limited. And people did experiment. They went there with the ship and they start to scoop iron in the ocean, iron dust. And suddenly all the water turns green because they were just lacking iron to be able to grow. They had everything else they needed. So limiting nutrients might stop the reaction. They are actually theoretically able to grow very fast. Remember yesterday, all biogeochemical cycle, all element cycle are linked together, and cells require many elements to work. So if theoretically the reaction can go very, very fast, doesn't tell you that practically it's going, because other things might limit those cells. The most interesting things as an environmental engineer or geochemist or geologist or modeler of environmental things, that you should know that life uses a big part of the periodic table. Now, these are all the elements that life can use, all of them. And it's not a complete list because, for example, we just figured this out. Lanthanum, cerium, niobium, these are new. We didn't know life could use them. Now we know it can. But life can use tellurate, selenate, mercury, gold, cadmium, tellurium, uranium, you name it. It's easier to count the white dots than the other. And every few years, we add a new one that life can use. So you should assume that life uses a lot of the elements out there. Some of them are used as a building block to make things like carbon and nitrogen. Some of them are used as a substrate, like arsenic or iron or hydrogen. Hydrogen is both a building block because you do carbohydrate, lipids, CH, but it's also substrate, hydrogen as an electron donor. Some of them are just metabolized, just because they're there. We don't really know why, like mercury. There's a lot of microbes that can take mercury 0, reduce it to mercury 2, methylate mercury and spit it out as methyl mercury, which is the most toxic form of mercury out there. Why they do it, we're not sure, but there's a lot of microbes that do it. So microbes interact with elements for a lot of reasons, not always clear, but they do with a lot of them, okay? And to me, it's impressive to see just how much of the periodic table they can actually interact with. And they interact with minerals. That's another thing you should keep in mind. Now, these are 3 very basic examples of minerals that microbes help to precipitate. The first one is amorphous calcite. Microbes are very, very good at doing calcium carbonate of many types. Calcite, aragonites, and very different forms, dolomites, and so on. Second one is pyrite framboids. That's weird structure at the center. That's usually sedimentary pyrite. That's usually hallmark of microbes doing sulfate reduction. And then the H2S produced is mediated with the iron present, and you get this beautiful framboid structure. The last one on the right is iron hydroxide, rust. There's a lot of microbes that when they precipitate, they do iron oxidation, iron 2 to iron 3, to avoid getting covered in rust and getting stuck. They build a twisted stalk, a ribbon of iron hydroxide. And you see it there. And there's a cell there that is actually building that. And microbes interact like this with many minerals. This is a short list of some of the minerals. Goethite, lepidocrite, ferrierite, magnetite, hematite, margimite, siderite, vivianite, I don't even know what that is. And you can see this list. the point here is that it's not just a long list of minerals they interact with, both dissolving, giving electron directly to the solid mineral or taking electron, or precipitating it. This list is made of mineral we have tested in the lab. Okay? But what I told you yesterday, how good are we at growing microbes? Very bad. Less than 1% on average. So our knowledge of the mineral microbes interact with comes from a very small group of microbes we can grow in the lab. Reality is that out there, microbes might interact with a lot more mineral and we have no idea. And we know microbes that don't need the hydrogen as electron donor. You stick an electrode there, you touch current, and they get electron directly from currents. They don't have, they're perfectly active. And they dump electron directly on any mineral that can receive electrons. So even if you go, and this is part of a very long list of minerals, remember that comes from our view in the lab, which is a minimum portion of what's happening out there. So microbes interact with a lot of elements, doing any sort of chemistry, including a lot of redox, and microbes interact with a large number of minerals. And the list is growing every year. We figure out new minerals, new molecules, new interaction. Now, if you want to do a list of electron donor and acceptor, it's easy. You take all the two species you can think about, and you put them in a line. And then you take all the oxidized species you can think about, you build the second line. And then all the intermediate species, you put them in both places. Like, for example, elemental sulfur, right? Can be a donor and an acceptor. Thiosulfate can be a donor and an acceptor because we are in that mid-range. You can both oxidize and reduce. But the list is very, very long. And we don't know all of them. And we discover new electron donor and acceptor that are possible. And all the combinations are theoretically possible as long as delta G is negative. But we are not sure if they're all used. A colleague some time ago wrote 1,500 redox reaction involving only hydrogen, carbon, oxygen, nitrogen, sulfur, and iron only as two minerals. He wrote all the possible combination, 1,500, calculated the general delta G, they're all negative, then calculated delta G for ocean water, for groundwater, from geothermal environment. And they're all negative to different extent, right? And then you look at this very, very long list that does not contain strange stuff like selenium, arsenate, uranium, tellurium, all the weird stuff. Only the most common stuff, 1,500. And we don't know how many of these are actually used by microbes. We don't know who's using them. There are still very basic questions we cannot answer. And if you add only one more element, like arsenic inside, you're going to add 3,000 more reaction because it's going to combine with those 1,500 and things become gigantic. And simple question like how many of these reactions we know? We don't, because they're theoretically possible. We have evidence that some microbes do it, but we don't know who is doing them in the environment. So there's still a lot of fog in this world. Now let's do one exercise together. I'll take this one up here. H2S plus oxygen gives you elemental sulfur. That's sulfide oxidation to elemental sulfur and is aerobic. Why aerobic? Every time there's oxygen, we consider it to be aerobic. If you are in other conditions, which are not specified, often you are in anaerobic condition, lacking oxygen. So that one we will call it sulfide oxidation to elemental sulfur, aerobic. I'll take another one. I'll take the third one from the top. That's ammonia oxidation, aerobic, to nitrate. And three, right? Anybody wants to try to read the second to last? Any volunteer? Come on. Go on.
Iron 3, iron 2, oxidation, aerobic.
So that's iron oxidation, aerobic. Very simple. That's the name of one metabolism of a set of microbes in the environment. Anybody else wants to try it? Anyone? Let's try this one. Anyone? Come on, we'll do it together. Don't let me volunteer you, volunteer yourself.
Reduction.
Sulfate reduction
Okay.
to sulfide. Hydrogen oxidation, because there's hydrogen there as electron donor. Hydrogen oxidation coupled to sulfate reduction to sulfide, right? Try the one just below.
Elemental sulfur reduction. Coupled with hydrogen.
Still oxidation. Hydrogen always get only oxidized, right? Hydrogen is only with only an electron donor. So that's elemental sulfur reduction to H2S linked to hydrogen oxidation. Try this one. Try this one. Anyone? Aerobic methane oxidation. That's how we call it in microbiology, because methane is always oxidized to CO2 in biology. So aerobic methane oxidation. There's a few more we can do. But once you get the slide, try. You're basically calling out metabolism of microbes. Those are both abiotic reaction that can happen in your system or some system out there, and they're potentially carried out also by microbes. So next time somebody tells you, we're looking for sulfate reducers, you know exactly what we're talking about. Next time somebody's talking about aerobic methane oxidation, you know exactly what the microbes are supposed to do. Next time we'll be talking about ammonia oxidation in a reservoir. You know exactly what we discussed. Okay, this is relatively, once you learn, it's relatively straightforward. When you look at this reaction, what you're looking at is always electron donor and electron acceptor, right? We're not saying anything about the carbon. You remember the triangle. You need a donor, gives you energy, the electron. You need an acceptor when you dump things at the end, and then you need the carbon. We're not telling you if any one of these microbes what carbon is using. Okay, we'll get into that in a second. So what we said so far, we have two types of energy sources on this planet, chemical and light. They both boil down to redox chemistry, but we have specific name for microbes and organisms that use one or the other. Anybody using chemical energy is called chemotrophic. Trophic from eating, is eating chemistry. Anybody using light is a phototrophic. Phototrophic organism. Phototrophs, for short. So that's the basic distinction. And then you can go a bit down. So this one is telling you if the primary energy is light or chemistry. Then you can go down. And of course, organic chemical energy can come in form of organics or inorganics. And we have a distinction. So chemotrophy becomes chemoorganotrophy if it's organic, or chemolithotrophy from lithos, rock, if it's inorganic. And the phototrophs, they stay phototrophs most of the time. So very often, when we speak about the metabolism before that, we say, okay, this is a hydrogen oxidizers, there is respiring nitrate. So it's a nitrate reduction to ammonia linked to hydrogen oxidation. Remember this one, hydrogen oxidation, nitrate to ammonia, this reaction. Now, the next word that microbiologists will say, oh, these are chemolithoaldehyde, meaning they're using chemical energy, inorganic, and then carbon. Or they will say these are heterotrophs, meaning they need organic carbon. They're on the chemorganotroph side. So what we're talking about at the end, we said where the electrons are coming, hydrogen oxidation. We tell you where the electrons are going, nitrate to ammonia reduction, and then we say what's the carbon source. Biologists are constantly talking about these three things because this is the basic you need to understand the metabolism. Because if your microbe is a chemolithotroph and is using CO2 as carbon, well, your cushion gas provides the organic carbon, the carbon to build things, right? If your microbes is a chemorganotrophs doing the same donor electron reaction, but wants organic carbon, and in your reservoir, you don't have any sugars, any lignin, any cellulose, no organics, only CO2. Well, you say the reaction is not going to go anywhere because your microbes want organics as a carbon source. So you can use this to your advantage because, as we said, you need electron donor, electron acceptor, and a carbon source. So every combination of these different things is valid. Photo, the energy form is light. Auto and hetero tells you only if the top one, the carbon source is inorganic carbon or the carbon source is organic carbon. And then litho and organo tells you if the electron acceptor and donor are organic or inorganic. So you can do photolitho, autotroph, chemolitho, autotroph, chemolitho, heterotrophs. We are chemo, hetero, organotroph. We use chemical energy. We want organic carbon. We use the organic carbon as carbon source, hetero, and then organo, because it's the organic carbon, the electron donor, trophs. Chemo, hetero, organotrophs. Plants out there are photolitho, autotrophs. So we use this as a general term. Less important for you, but it might come up in discussion. It's fairly easy. So I'm going to mention a few general metabolisms now, which I think becomes relevant tomorrow when we discuss. Photosynthesis is a general term for any light-driven metabolism. Now, the photosynthesis out there is called oxygenic photosynthesis, the one that plants do, because they do they use light for photon, and they discard oxygenic, maker of oxygen. It's one type of photosynthesis. There are many other types of photosynthesis. If light is involved, you use chemical energy to do stuff. So there's other things like an oxygenic photosynthesis, where the electron donor is not water, like for plants. Plants split water to produce oxygen. That's where they strip the electron. But there are other things. They use light to strip electron from H2S, from iron 2, or from hydrogen. Electron donors and light is used to supplement the stripping of electron from those. And oxygenic phototrophs. Now, oxygenic photosynthesis is very important out there right now. It's the primary source of organic carbon on the surface, right? Plants are the primary producers around. And the ocean, not plants, algae, but it's the same. Now, the counterpart of photosynthesis is heterotrophy. Heterotrophy is any organism that is using organic carbon as a carbon source. You are heterotrophs. All animals out there are heterotrophs. So when you say heterotrophy, it's literally the use of organic carbon sources. So anything that requires sugars or any organic carbon is an heterotroph. Now, heterotrophy doesn't tell you what you are respiring, just tells you that your carbon in that triangle is organic carbon. Now you can respire oxygen as we do, but many microbes that are heterotrophs, they respire sulfate, they do sulfate reduction. When they respire nitrate, they do nitrate reduction. When they respire tellurate, they do tellurate reduction. So being heterotrophs has nothing to do with where your energy is coming or going. Only what your carbon source is an organic carbon source. The opposite of heterotroph is autotroph. Tells you that your carbon is inorganic, is CO2, or carbon monoxide or bicarbonate, but mainly CO2. So autotrophs, you are using inorganic CO2, inorganic carbon, heterotroph, organic carbon. Basic division. Yes. Like you can learn it by memory. Autotrophs, inorganic carbon source, heterotrophs, organic carbon source. And you can go back to this, and for each one of those, you can then ask the next question. Okay, this is an ammonia oxidizer. Is it autotroph or heterotroph? Meaning it's going to use CO2 or wants organics. And you can ask it for every organism, because in the triangle you have donor, acceptor, and carbon source. So you can ask that question multiple times, right? Every time. So heterotrophy using organic carbon source as carbon. And carbon is interesting, right? Because carbon has two different aspects. Carbon is the carbon source to make things, to make sugars, make proteins, make things, but it can be also an energy source or acceptor. Like we said, that methane is a good electron donor, is a reduced carbon. But that carbon is going to also be used to build things. CO2 is a good source for carbon to make things if you are an autotroph. But CO2 can be also a terminal acceptor of your electron, because it's an oxidized compound. So carbon has this dual role can play in life. It can be only the carbon source, or sometimes can play also the electron donor and electron acceptor, depending on the form of carbon. And look at this. These are type of carbon that life can use. This is what we call C1 compound, single carbon compound. Carbon monoxide, formaldehyde, formamide, formic acid, methane, methanol, methylamine, methylalide, dimethyl ether, dimethylamine, dimethyl sulfide, trimethylamine, and so on. The list is infinite. all these compounds can be used as carbon source by microbes. All of them. They're all small single carbon organics. They're all gained for microbes. Then you get other things that are gained for microbes in terms of carbon source. Aliphatics, aromatics. You can take all the toluene series, all the aliphatic series, all the aromatic series, all the phthalate, phenol, polyacetate, benzene, ethylbenzene, benzoate. They're all used by microbes as carbon source. No problem. All of them. So when we say organic carbon, generally people refer to sugars, but it's not just sugar. It's literally almost any type of organic carbon. There is a microbe that can actually take it, break it, derive carbon pieces to build stuff inside the cell. So keep that in mind, because in reservoir, for example, you often have residuals of hydrocarbons, because that was used as a methane well or oil well in the past. So while you don't have source of sugars, you still have a ton of organic carbon that microbes might exploit, right? And they might exploit it as a carbon source to build things. We told you that reduced carbon can also give electron for reaction, right? It can be an electron source. So you can do things like get electron from methane, or get electron from methanol, or get electron from formaldehyde. So you get both carbon and electron. In your triangle, a single molecule covers two of them, right? And then you have a special type of metabolism, which we call fermentation. Which fermentation, essentially, that triangle, you have donor, acceptor, and carbon. I just told you that sometimes organic carbon, reduced carbon, can be both donor and carbon source, right? So we are collapsing two sides of the triangle in a single molecule. In fermentation, things collapse completely to itself. So microbes will not use an external electron acceptor like oxygen to dump things, but they will take a molecule, break it, take an electron, use a piece of the molecule as carbon source, break a little more that molecule, and use that molecule to dump the electron back in. So for example, happens with sugar. Sugar gets reduced all the way to pyruvate, and then pyruvate, you dump the electron inside and you make lactic acid or acetic acid, or one of the many things we enjoy in fermented food. So in fermented food, you don't have an exogenous substrate as respiration. You don't dump electron on another molecule. All the triangle collapses on itself. Why this is relevant for you? Well, fermentation, which is taking organic carbon and doing an entire process within that set of molecules without external acceptor, happens a lot, as you may imagine, in anaerobic system, right? Where there's no oxygen and all other electron acceptor, nitrate, sulfate, have been depleted. There's nothing around there. There's only organic carbon. Microbes are good enough that they can break it and make extra things and dump them out. And one of the byproducts of fermentation is hydrogen. One of the ways in which microbes get rid of some of the electron is get protons, dump an electron there, put two protons together and release hydrogen. So biological hydrogen is produced by this pathway here. So when you are in an aerobic system and a lot of other good oxidants are depleted, and you have a lot of good organic carbon, microbes produce hydrogen by fermentation. That's why it becomes relevant for you guys. It's a contributor to hydrogen in the cell. And for example, in some porous reservoir we analyzed, we see a lot of microbes. There's no hydrogen there because there's no production hydrogen well. There's potential for the future, but we see a lot of fermenters being present. A lot of microbes that are using the leftover hydrocarbons present, and they're fermenting them, and they're probably contributing to produce some hydrogen in the reservoir already, without us pumping it down. Now, as I told you before, where the carbon is coming from, autotrophic, inorganic, heterotrophic, organic, doesn't tell you what you're respiring. You are heterotroph, but you also iron, you're using oxygen. There's plenty of microbes that are heterotroph like you, but they don't use oxygen. They respire nitrate, sulfate, other things. Because we are oxygen dependent, humans, we like to make a big distinction. If you use oxygen or use everything else, we call it aerobic respiration using oxygen and anaerobic respiration using everything else. So it's a big lump of everything else. So that's a huge distinction, right? So oxygen to water, aerobic respiration, one of the most powerful oxidants available on this planet. If you're using manganese, iron 3, nitrate, nitrite, elemental sulfur, sulfate, thiosulfate, and so on, you're doing anaerobic respiration. So every time you go back to the long list we were looking at, here, every time somebody is using oxygen in the reaction, like these guys up here, you know they're aerobic, they're using oxygen. Every time they're not, they're using nitrate, sulfate, thiosulfate, elemental sulfur, tellurate, arsenate, they're anaerobic. In subsurface ecosystem, the majority of your metabolism will be anaerobic because we know oxygen is not present in abundance unless you inject it by mistake, right?
In the case of hydrogen injection on the ground, we can also include oxygen. So they were going to change the environment.
Yes, and how much oxygen you inject is going to really make a big difference, right? Because if you're injecting a few parts per million, it's going to get consumed very quickly and not start a big community. If you're injecting a percent amount, then you're giving off your flank to microbes doing all sorts of crazy stuff, including oxidizing elemental sulfur and pyrite and making sulfate that then can be reduced back to hydrogen sulfide, the micro your system, freeing iron 2 that then becomes an electron donor, you set off a bomb. So knowing exactly how many things are using and how much, we'll have to make specification for what you're injecting now. And we know that cleaning a gas to 1% oxygen or 100 ppm oxygen or 100 ppb oxygen is millions of dollars difference in operations, how much you're cleaning your system, right? And so finding exactly those levels will be crucial to provide specific to the engineer and for the economic side of injection. I've been dealing with giving advice to companies that have been doing carbon injection underground, because how much contamination of oxygen is allowed together with CO2 injection makes a big difference for money, but also a big difference for the community response. So a special case of carbon as terminal electron acceptor is microbes making methane. Microbes, when there are no other electron acceptor, they can use CO2 to dump electron there. And they do it reducing it to methane on the way. So we call them methanogens. Now that's something that doesn't give a lot of energy. So you become competitive only when every other electron acceptor has been used around. Because if there's still nitrate or sulfate or iron 3 as electron acceptor, people that use it, they get more energy, so they overgrow guys that do methane. But methane is a big one underground. Now, just to give you, and this is more for you to read later, if you think about nitrogen metabolism then, so if you think about carbon metabolism, you have the heterotrophs using organics of every time, the autotrophs using CO2 to build things, and then methanogens that respire CO2 at the end. In terms of nitrogen, what you have? Well, you have, this is a general view of the nitrogen cycle, but what you have oxidized nitrogen species that are reduced by microbes, nitrate, nitrite, nitrous oxide, nitrous dioxide, and so on. And then you have reduced nitrogen species that can be used as electron donor, like ammonia, hydroxylamine, and a few others. So every time you think about what are the nitrogen metabolism in my system, well, the oxidized species will be used as an acceptor of electron. The reduced species might become a donor of electron. How much of this I have in my reservoir of each type? And then you can start to connect dots, right? The same you can do with sulfur metabolism. So sulfur metabolism, sulfate is one of the most important, but generally you think about, you can go up and down through the oxidation states from sulfide to sulfate and so on. But generally you think about oxidized sulfur species as acceptor of things, place where microbes can dump electron, and then reduced sulfur species that can actually provide electron to microbe energy source. And you can do this thinking for all of them. I just wrote carbon, nitrogen, and sulfur here. And then I added one more, which is iron, because I think it's very relevant. But iron, if you look at sulfur, look at this. You have many intermediate that can act both directions, donor or acceptor. Iron is trivial, but very important in the subsurface. You only have two species, iron 2 to iron 3. If you give electron, iron 3 to iron 2, if you accept electron. And so these are your two options, but iron may become a very good electron acceptor in the subsurface because you may have a lot of oxidized rocks in your reservoir or around your reservoir. A lot of the sandstone might contain oxidized iron in there. There is enough for microbes to use to dump electron. And at that point, your hydrogen is going to be consumed because microbes can dump electron on iron 3 on your rocks. So iron is an important one. And next time you go to get water from a reservoir, stuck it under a microscope. If you see twisted salts, you know your microbes are doing iron oxidation or they're dissolving iron one way or another. That's a good example. I left only one slide on this because we said autotrophs, right? They do carbon, inorganic carbon to build things like plants do. Now, we know very well about plants as primary producers, but we said before that a lot of microbes can actually use CO2 to make things. We call them chemolithoautotrophs. They use chemical energy, donor and acceptor are inorganic, so lithos, and then autotrophs, inorganic carbon source, which means that many microbes are also primary producers, like plants on land. They're the base of the food chain. They produce the organic carbon that everybody else is feeding on, herbivores, carnivores, by consequence, and the entire trophic chain. But this happens underground. And we call this carbon fixation, autotrophy. And there are many pathways by which microbes can use inorganic carbon and make sugars. I wrote here just the more important one. Calvin cycle, we call it this way, it's what plants do and many microbes and all the algae. Then we have a few more that are mainly found in the subsurface. The point is that, the key point here is that in the subsurface, you have some microbes that work like plants, in meaning that they are producers of inorganic carbon to organic carbon that everybody else can feed on. They're the plants of the subsurface. They do it without light, but they're still moving things from inorganic to organic. If you can imagine what you have in the subsurface, you have an entire forest ecosystem. You have some microbes that are primary producers using inorganic carbon. They use chemical energy and inorganic carbon to make sugars. And then you're going to have microbes that eat these microbes. And then you're going to get a complex food chain. So don't imagine your system as having, one microbe that does one metabolism. And that's what we care about. People are cross-feeding from each other. Your waste becomes my food. You become my food. When you die and you break and you free molecules everywhere, I'm going to be able to pick them up and use them like a scavenger. So there are very complex dynamics happening. And I'll show you a general reconstruction done for the ocean. Tomorrow we see things that are more relevant to reservoir. But one thing is doing this. So this is, the quality is not great, but we can do. So this is ocean from the top to 4,000 meters at the bottom. And we are in mid-ocean ridges where you have geothermal system at the bottom. And what this colleague did is, let's start from the top and let's think about what are the donor, the acceptor, and the possible metabolism. So when you are in the photic zone, the area where light can penetrate, you have for sure microbes using light to make photosynthesis. That's OP at the top. Water is your electron donor. And then you are autotroph, fixing carbon. And then you can also have an oxygenic phototroph using H2S. As you get deeper, what are the good electron donors there? Well, water cannot be used as an electron donor anymore because if you go deep, there's no light. So you don't have photon to break water. What do we have? Well, we might have ammonia, electron donor, electron acceptor is oxygen, or we might have nitrite, and then ammonia again, but with nitrate as electron acceptor, or sulfur species as oxygen, reduced sulfur species against oxygen and nitrate. We keep going deep. And then you go into the sediment, for example, and then you might have sulfide oxidation coupled to something else. H2 as donor, CO2 as an acceptor, and make methane. You can literally try to write down for everywhere you are what are all the possible donor and acceptor that are there. And that's the basic of understanding theoretically first, and then testing how your system works, right? So imagine you starting a drilling operation, and you know what you have at the surface, and then you know the stratigraphy, and so you know what compounds different rock might provide as you drill to your system. And then you know the composition of the reservoir, and you know the composition of your drilling fluids, and your injection gas and cushion gas, and so on. And you can write down every possible valid reaction that might feed your microbial community down there. Now, this would be your theoretical boundary of your system. And then from there, figuring out not just which one is theoretically possible, but which one is likely to happen, and then later which one is happening, gives you an instrument to interact, managing your system, managing risk, going into the injection in future, and all things like that. Now, nobody expects you as an engineer or a geologist or a physicist to do the job, right? But you being knowledgeable enough that you can see the problem and then contact a microbiologist, an expert, and what we did to figure this out, that's what we want from you. Because right now we've been approaching these in underground hydrogen storage and carbon capture storage and a number of other things completely separate. So it's like, you know, biologists do their things without thinking about what geology is there, what rocks, what things. And at the same time, geologists do their things like everything is abiotic, microbes don't really matter. And it's only in the last 10 years, maybe there will be an interactive moment. So you thinking about thermodynamic reaction, dominant acceptor, redox, and what are the things that might go wrong here, biologically speaking, requires you to interact with the biologist. Tomorrow morning, we'll focus exclusively, deep dive into hydrogen. Hydrogen metabolism, what microbes do with hydrogen, what they do underground with hydrogen, what's the relevance to underground hydrogen storage, natural hydrogen and carbon capture storage, and how you might interact with biology to try to figure some of these things out. Key message for the day, microbes use a lot of the periodic table. Every time you have a thermodynamic value, which means delta G negative redox reaction, microbes might do it, and the relative contribution of abiotic to biotic is the most important things in your system, and that's the most difficult things to figure out. Questions? Yes.
Yeah, okay, so going back to what we discussed.
Yes.
So this is, I guess, a super complex system because it's an open system and it's continuously renovating but in a reservoir where there hasn't been any, for example, influx of new materials, new organisms, things like that. It is a closed system and it is a system that has reached some sort of stability somehow, right?
Until you inject, yes.
So, how would, for instance, a microbial community that, for instance, requires hydrogen or things would have survived without that hydrogen? Why it could be a concern for, you know what I mean?
Yeah. Why should we worry about being there microbes that can use hydrogen there if we have not injected hydrogen yet? Well, where are we? Are we on a sedimentary basin in an area where you have also organic rich layers like coal or sapropel or other things deep down? Well, if they're deep down, thermogenic decay of organic matter creates hydrogen. So there might still be hydrogen coming up. Are we near a cratons, stable granite formation somewhere like Brazil, South Africa, Pennoscandian shield? Well, radiolytic hydrogen is big in many of these places, radioactive decay. At the end of the day, there are two things to consider, and we don't have a perfect answer. First one, you say stable microbial community. What does mean stable? Stable over which time frame? Because for an ant born in the winter, a tree without leaves, that's a stable tree. But then the ant doesn't know that in spring this thing is going to bloom up, because the time frame in which he's looking at the tree is a dead tree for his entire life. So if your concern is about stability of the microbial community over weeks to year, yes, probably stable. If that stability you push it to hundreds or thousands of years, we have no idea. Nobody ever looked at variability at those scales. Plus, we know these systems are stable up to a point. These systems are fractured. We have no idea if they have long-term recharge of aquifers at the bottom. So they're closed system, depending on the time frame you're considering, right? Like the planet is a closed system if you don't consider sun energy coming from the sun and hydrogen and helium being lost to space, plus a small amount of stardust coming in with meteorites and things like that. But time scale will tell you a lot, or if you're considering really a closed or an open system. The other thing is that I think worrying about reaction in your reservoir, it's dependent on what type of system you want to use. So if you are injecting hydrogen there to pull it up in 6 months and then burn it and then inject it back and up and back, and you want to do a total of 5-year cycle for your reservoir, you have a set of problems. If you want to inject hydrogen down there and then keep it there for the next 25 years as a strategic resource, then you have a very different set of problems, right? So using the small scale reservoir for weekly to monthly use of the injection production system has a very different set of problems with microbes than storing hydrogen in a very large reservoir for strategic use down the line. And those two things will require you to think in a different way about stability, about closed or open system, about reactivity of the community. In microbiology, for a very long time in the past, people have been thinking about the distribution of microbes on the planet. And they've been puzzling their head because in the ocean, you know, if you take hundreds of liters of seawater and you filter it and sequence the DNA, and you count every possible microbes in that hundreds of liters of seawater, you're not going to see a single organism, at least we haven't, that is capable of degrading aliphatics oil, petroleum. And yet, if you drop 1 liter of petroleum out in the ocean tomorrow, within hours, thousands of them come up and start to eat the petroleum. Where are they coming from? They're not appearing out of thin air. Are they coming from far away? But connectivity is not that high. Are they there in very small abundance and dormant for tens of years? And then when a drop of organics comes up, they just bloom. So people used to say, everything is everywhere, environment selects. Meaning that there's a certain degree of connectivity where all the metabolisms are dormant everywhere. And then when you trigger the condition, they come up. Now, this is still debated in microbiology. We're not sure if this is the case or not, or there's some type of longer term connectivity. But I can assure you that if you go in a reservoir where there's no hydrogen present, or you don't see hydrogen present, you pull up microbes, you provide hydrogen in the lab, they bloom in the lab. In many places, probably, if you go in marine sediments, you dig down like 1, 10, 100 meters, and you measure free hydrogen is nanomolar to picomolar concentrations, very low. But if you measure how many microbes are expressing proteins to use hydrogen, all of them, we call them cryptic cycles. One guy is producing hydrogen that is used by him, the other is producing something used by him that is making hydrogen that is used. So there's zero accumulation of free hydrogen, but everybody is passing hydrogen around. And we discuss tomorrow, hydrogen is the most versatile electron donor for biology. Everybody can use hydrogen almost. So even if we think there's no free hydrogen there, microbes are still shuffling hydrogen with each other like a currency, like it's US dollars. They just go around all the time. So I think we need to stop asking general questions like what we do with reservoir and microbiology, and say what we do with reservoir and microbiology for this type of work, for that type of work, for long-term storage, for short-term storage, and so on. Yes.
How fast can microbes adapt for people with changing environment conditions?
That's another very good question. So microbes evolve in very different ways. One way is just by slow changes in their DNA that get selected, generally random mutation or errors of making a copy of themselves. The other way is by passing around information. Microbes can take a piece of DNA, pack it in a small round molecule, which we call plasmid, which is like a floppy disk, that then you pass to the next microbe next to you, and the microbe gets a new function, gets a new problem. And that's the way, for example, antibiotic resistance goes around. Microbes pass around a small piece of information on how to survive the use of antibiotic. The reality is that we have evidence in the lab that microbes may evolve very fast, like days to weeks, and evolve new function and adapt to new condition. At the same time, we have no idea how this happens in natural environments that are far away, or how common, or how fast it is. There is suggested that it's pretty common. I'm going to show you one video, just 1 second, just to show you how impressive this can be. See if I can do it here. Let's try if I go hotspot with my. This one. So in this experiment here, what my colleagues did, they've been taking a very, very large table and they put these gel-like things in which we grow microbes, microbe grow on it. This is a nutrient, simulates the environment and microbes grow on it, okay? But what they didn't do a single layer. They did 2 corners, and then the next layer, they put antibiotics in it to a concentration of 1. And then they made another layer where the same antibiotics, then a concentration of 1 killed the microbes, they put it at 10x the concentration. And then they did the central layer to 100x, or even 1000x, I don't remember. So basically, you see they start microbial culture on the side, the microbes grow, they get to the border where the killer antibiotic is present, and they stop until in hours or days, 1 microbe will evolve a situation and will resist our antibiotic, grow again, get to the 10 times more concentrated and stop. And then within days, somebody's going to break loose again. And they keep going until they colonize everything. So 2 by 4 feet is 2 meters by 60. 1.5 meters. Look, different layers. And then they put agar that had 0 antibiotic. And then they increase the amount. So 0, 1, usually that's enough to kill them. 10, 100 times more, 1,000 times the concentration for the killer. And then they pour the bacteria and they let things see how they play out. And now look at the real time. These are the microbes growing. And they get to the border there. Then suddenly you see that somebody, poof, look at that, comes out. It's colonizing the 1x and multiplying. That's a new population adapted. We talk in days, and look how many mutants appear. And they keep going until they are at the border of 100 times the concentration, which is deadly for all of them, absolutely toxic. And then somebody breaks loose again. Somebody already passed on that side, and now on this side, multiple population emerge. And then they repeat this at the 1,000 times concentration. So 10 to 100. In 11 days, they make it to the 1,000 times the lethal dose they had before. Okay. This is how fast they go. And every point they draw here is the initial one, and now mutated. From initial 3 inocula, population spread, find the mutation, keep going, keep going, keep going. Evolution in action. 1,000 times the concentration we were using to kill them, adapted in 11 days. Yeah, real-time evolution. One of the many real-time experiments that have been done.
What does this colors mean?
Oh, they're just using color to show you different generations of microbes. So the red one is for those that break into the 1,000 successfully. So on the other side, only one did. The orange one got that, but they never survived. So they're just tracking evolution. They're using that to calculate rate of changes. And I bet that every one of these lines that solved the problem of how to grow from a 10 times to 100 times fold, found a different solution to the problem. So those are all independent evolutionary events, because you can solve the same problem in many ways, right? And this is only 11 days. So microbes can adapt pretty quickly. Now, unfortunately, we don't have experiments here, reservoir or anything like that. The last thing I want to mention is also viral infection can help microbes adapt, because when viruses go around, they carry genes. And if they go dormant, like the cold sore in your mouth, they sit there, you have extra genes there, and they might pop out and be used. So viruses are like USB stick with extra programs there, and sometimes they get stuck in a computer and they give new function. So it's very fascinating and complex. Yes.
Yeah, you were talking a lot about now about culturing and everything, but what is the problematics in situ monitoring?
No, it's methodological. Let's make an example, okay? Let's say you want to measure the rate at which microbes in a closed source system are using hydrogen, okay? Now, you cannot go down there and make measurements, right? Let's say you can actually pull up the sample without changing pressure, temperature, pH, nothing. There are ways, very complex, very expensive, but there are a few ways by which you can get water 3 kilometers depth, bring it up, not depressurize, not do anything, right? You have it in a perfect capsule that is not exchanging anything with the environment, which is already very difficult. Most of the cases are degas, changing pressure, changing temperature. Let's say you can keep it at the same PT, pressure, temperature, everything. And you can do measurements there. Now, you want to measure how fast they're using hydrogen. How are you going to do it? You're going to measure them while they're using hydrogen that is already there. Well, if they're using it, and you are now in a small closed system, the hydrogen is going to go down very, very quickly because it's used. So if the speed of a change in reaction is the slope tangent to your substrate going down, as they use the hydrogen, your slope is going to be like this. But in an open system, in a system that is big down there, your slope might be this, because hydrogen doesn't become limiting immediately. The alternative is that you inject hydrogen in your small system and then you measure how it's going down. But now you are artificially increasing the amount of hydrogen because you're providing substrate to be able to measure it. And so you are getting the maximum theoretical speed when they have excess hydrogen. But in situ, they don't have excess hydrogen yet because you're not pumping them. So the point is, every time you are trying to measure something, you are manipulating the system in one way or another. And every time you're touching the system, you don't get what they're doing in situ, you get what they're doing under the condition you use. And all the technique we have to measure things, screw things up. For example, people measure hydrogen by gas chromatography, right? You have your closed system, your tiny bottle, you have your formation water from your reservoir, your headspace with hydrogen, you're doing incubation, you're measuring every hour. How do you measure? Well, I go inside with a syringe, I get 1 ml and inject in GC. Yeah, after the first injection, my volume here just went down by 1 ml. If I measure every hour, in 2 days, I will be removing 45 ml of headspace. That's going to change pressure, which changes the solubility. I'm going to remove both product and reactant, which is going to affect how the chemical reactions are. What do? Do we add more hydrogen every time I pull hydrogen out? Well, they have always excess hydrogen, but they're accumulating byproduct there, which is going to slow the reaction because product accumulate on the other side. Anything we try to figure out right now gives us something that is a measurement, but it's not necessarily representative of it. is similar to what the problem that people have with, you know, when you start to interact with electrons and quantum mechanics. The only act of measuring changes the system in a way that doesn't really represent how things are going. You see the experiment with the electrons in the tunnel, wave and particle, depending what you're doing, you measure things. In this case, the act of interacting to measure something messes up the system. Microbiologists discuss usually theoretical maximum rates under a certain condition. Nobody will ever say in situ rates because we know that things screw up. And hydrogen, you can do it by GC. In other case, we provide substrate with the heavy isotopes or radioactive isotopes. But in that case, you are pumping in excess substrate for them to take up. And it's complicated. One of the things I've started discussing and thinking and doing very soon, hopefully, is to do high frequency measurement of hydrogen disappearing in a system, in closed system, without GC, but with Raman. So you can measure Raman spectra of hydrogen every 20 seconds or 30 seconds. And so you can start to get depletion of hydrogen when hydrogen is not depleted yet, but is at the start of the work. But you need to do it within seconds to minutes of collecting the sample. Otherwise, if you wait an hour, the hydrogen is being used. So complicated, technically, not theoretically, technically, practically.
Okay, Donato. Let's do the way around, because I understand that the in situ sampling is complex. My question is that one, how much, suppose you take a sample from the subsurface, you put it in a kind of system which is a pressure temperature equivalent with a similar type of nutrient. My question is, what has to be the minimum dimension of your basically
Unit.
modeling simulator with real blocks that can approach the nearest what's happening in the subsurface? My question, I mean,
We don't know.
what's happened in the microbial system? If some of them are isolated, if you're isolated 1 kilometer cube, or it's 1 centimeter cube, is it going to change the behavior of microbial? Yes, no.
Yes.
How far?
Well, if you're assuming your 1 kilometer cube reservoir conditions are absolutely homogeneous.
Yeah, with no fluid interaction because you have a ceiling above, you are not touching anything, the only introduction.
You can leave. If you consider they're absolutely homogeneous, and there are no difference in rock composition, we don't know because nobody ever did this scaling down. But I will tell you that liters to meters, so thousands of liters would be more than enough to simulate stuff. Even at 1 liter, you know, to 1 to 5 could be enough. The problem is that in the majority of the case, what we miss is the heterogeneity of the system. That's what we miss. The smaller you run your experiment in size, the less heterogeneous it is.
Okay.
That's where we lose. But we lose that in seawater as well. And another thing that we see in seawater, so people have been doing these experiments in seawater for a long time, right? You have thousands of millions of liters of seawater. You want to know what happens? You close it in a container and same temperature, pressure, light, everything, and you try to see how the community behaves. Deep sea, so there's no atmosphere, there's nothing, right? A capsule. The problem is that, for example, the surface of the glass or whatever container to volume ratio is going to create a discontinuity there. There is no presence in the system. People talk about bottle effects. Microbes behave differently because now they have a boundary they can attach to. It doesn't exist out there. So they figure out that if you do an experiment in a 1 liter bottle, or do an experiment in a 1,000 liter container, the results are not the same, even if theoretically there are no heterogeneities, just because the volume to surface of contact ratio between the two changes.
So what are the first sort of magnitude parameter which control those things?
Exactly, we go back to first principle, which is what we're trying to figure out. I can tell you what are first order of magnitude controls in the ocean, because we've been doing ocean microbiology for the past 70 years. But we have not been doing porous media microbiology for 70 years. So the questions are still all open. We have no idea. What's the primary control in the subsurface?
Yeah, let's say like for a specific hydrogen project, we did the like biological study, and we know that like at some places there are risks of microbial activities. What are the risk management strategies that can be?
this is another very good, very interesting question. So right now, people in the oil and gas have been already dealing with microbial problems, namely the souring of wells. So you have a well that is in production, something happens that your well starts to produce excess H2S. Removing H2S from your gas is expensive. And sometimes the problem extends quickly to the entire reservoir because it's a chain reaction and it doesn't become as profitable to put things out. So people have been dealing with souring in a number of ways, using biocide, injecting things that will kill the community, slow them down, bind the H2S and so on. So there are strategies to interact with it. None of this is perfect. None of this works 100%, but there is a big literature out there for souring, for example. Now, we're not suggesting that the exact same approach should go because we're talking about different microbes and different metabolisms, but we could for sure interpret and adapt some of the strategy used for microbial management of oil and gas wells to underground hydrogen. There are a few additional things we're still trying to figure out before we get to that. How you design a monitoring strategy to know if your reservoir has a microbial problem. Well, if they're producing H2S, it's easy because you start to pull up H2S with hydrogen. But if they're precipitating pyrite and they're clogging your pore space, how long is it going to take before you realize you're losing space and you're losing hydrogen down there? Is there any other way we can figure this out before it happens? And I'm making just the example around pyrite and this, but there can be a number of other things that are happening down there, right? So we have our ultimate goal is to monitor and manage microbial risk of production wells, right? But before getting there, there's a number of things we need to figure out first. What metabolism are possible, how likely they are, what are the rates at which happening? Because if the rates are super slow, because they are carbon limited, phosphorus limited, whatever limited, and our injection strategy lasts only 20 years with monthly production or 6 months production, maybe we don't care. We don't need to care. And then what's our monitoring strategy? And then what's our intervention strategy? But that's the ultimate goal, absolutely. It's just that right now it's so far down the line that we're starting to discuss possible strategies.
Still a strategy that we surpass this process and let's say like for all of them there's a strategy. If there is a risk, we can do this and we don't.
Well, my first suggestion would be don't build where you have a high risk of microbial work. Just let's pick a reservoir where conditions are unlikely for microbes to start to produce things you don't want. And let's not inject in shallow porous reservoir near sea where we have a lot of sulfate coming. Let's inject where we are at the boundary of temperature gradients that we know might protect the reservoir. And then, you know, that's we can do this together, you know. If we understand where the risks are, it doesn't say that we need to do that injection. We just move the injection somewhere else. There are political constraints, there's economic constraints, there's feasibility constraints. Let's add microbiological constraints to the mix, and then let's model what are the best reservoirs to do this. I mean, if you want to inject in the best place for microbes to do sulfide, and then you want to try to manage it, well, you're assuming high risk to start with, and that's on you. Why go there? Yes.
Actually, from this lecture, the most thing I learned is while you are talking about the condition that this kind of reactions can happen a lot. And I have one question for David. So I would say that the situation you mentioned is kind of we can define it as a representative elementary volume for the experiment, and also how to link with the numeric model. Because numeric model seems now, although we have good computational power, but we still cannot go to that kind of nanoscale. I mean, the scale is too small. Then we need I mean, the computational is not reachable. But so just like fluid flow, can we in fluid flow we use the permeability or porosity, this kind of simple scalar value to evaluate this signal cubic block. and how to link with the pore size. For example, if we have, for the experiment, if we have a pore sample, how to link with the pore size, pore general, and with the size of the pore samples, and meanwhile we eliminate the boundary condition because we need a system to close this shape of the experiment. So this can be linked with the surface area, the boundary of this experimental setup. But for our crosswalk, inside of the crosswalk, the surface area can be maybe 10 times or 100 times more. So, consider this kind of effect. Can we use the simple value to define, link with, first of all, we define the size of the full sample and link with the standard value to evaluate just like community. And meanwhile, the boundary condition of the experiment setup can be eliminated.
You are completely right, and some people have been doing this in some system using reactive transfer modeling and plugging the chemistry and the rates of microbial reaction within the set of equations that are used for reactive transfer modeling to model fluid flow in porous media. And it's been done. The problem is that the best we can do right now in doing this, and it's not a computational part of the problem, there are some models that have been developed, is that when we plug our microbial value for what microbes are doing, is either we have no idea what they're doing in terms of kinetics and rates and so on, so we plug artificial values, or our boundary condition for those values are so far away from each other that everything is possible. And you know, in modeling, if your pore space and surface area is not within a narrow range, but it's everything from 0 to 100, you can get any results you want, right? So there are theoretical models we can use right now to do some of this work. But the problem is that we need to step back for 1 second and try to get narrower range of microbial parameters to provide the people doing the modeling. Because at point we are all over the place. And we go from, don't worry about microbes ever, to, oh my God, microbe will eat all your hydrogen in 1 year. And that's not good for anybody. One thing we've been trying to do on our side has been categorize and put together all the possible theoretical reactions, right? And then Guillermo with Martina and with Bernardo, they've been trying to calculate delta G energy in situ to prioritize which one are highly energetic and low energetic, right? And then my idea was to, since we don't know in situ rates by microbes, but for many of these reactions, we know the abiotic rates in this condition because they've been experimentally measured, build a chemical reaction network of all the redox because one feeds into the other. And we actually brought this inside the new project we just got funded by Europe. Build a chemical reaction network of redox chemistry, plug in abiotic only rates, and then use that network within the context of reactive transfer modeling to simulate a purely abiotic system, right? So if you inject hydrogen, this concentration, this amount, and you run it for X amount of time, where you expect things to happen abiotic. Now you get an output, right? Where you expect the sulfate to go to sulfide only this percent, and ammonia to be this percent. Now you go back to your reservoir, that you just model, you take a water sample, and you measure the chemical species. Every time you're not matching your expected abiotic results, you mean biology is acting on that one. So you can actually constrain cost reaction rates by comparing a purely abiotic model against real life. This is something biologists did in the past. Some of the ammonia reaction we know today, they were completely unknown 20 years ago. People were measuring rates in digesters, big anaerobic digester, and ammonia was not there in excess, and N2 was there, and they were saying, what's going on here? None of our abiotic model is modeling this. Maybe biology is doing something we don't know. It took 20 years, and now we know exactly who's doing what, what's the organism, and how much they do. And I'm proposing a similar approach, which instead of waiting for biologists to characterize every single biological reaction to provide information to modelers, let's do the opposite. Let's assume there are no microbes, there's no life, there's no problem. But let's be good at modeling the abiotic redox reaction rates and so on in the context of first just as is, and then reactive transform modeling, and then we can compare that with real life cases and see where things don't match. We just proposed this. The problem is that I'm not a modeler, so I need help from you guys. And anybody of you that thinks that can help us to develop a reactive chemical network of redox reaction, more than welcome to contact me and let's collaborate on this. Otherwise, I'm gonna be stressing Guillermo to learn how to do it all the time. I think you're completely right. We can remove some boundary problem. We can model some of this. The problem is that how do we get good constraints of the different? There are techniques out there for the modeling. And computational is not an issue right now. The issue is more theoretical. And that's why we were discussing first principle. We don't need to model every single variable there. What are the important variables? Seems like pressure has a huge effect only on delivering in the liquid phase hydrogen, CO2, and the other gases, because it's a dissolution problem, right? Increase pressure, increase dissolution. But for microbes, the speed of reaction doesn't change a lot with the change in pressure. Other parameters appears to be more important, at least for the few reaction I know very well. So we need to figure this out together. And I think at one point within the project, concept of project, having meetings between modelers and microbiologists to discuss how to try to apply some of these things to models and start with toy small model and then scale up will be actually very beneficial. And that's what I hope to do also with the cost. Any other question? We can discuss more this afternoon or tomorrow morning. Yes.
Why? Maybe you've already gone through this yesterday. Why is it an issue for hydrogen storage and it's not that of an issue for metering?
Well, natural gas storage, as we were saying, we're not pumping anything. Well, we're not pumping the gas down usually, right? We're removing it.
No, I mean natural
For
gas
storage.
Storage.
Gas storage.
Okay. My feeling is that in some of these cases, the subsurface is limited by electron acceptor. When you pump down methane in large amounts, some of it is used, but it's minimal because they don't know what to use. My feeling is that hydrogen is going to be the same. I discussed this with David a lot of time. A lot of people scream on publication, microbes, we need all our hydrogen. I'm not sure that's going to be the case, unless we are in very specific condition where there are a lot of electron acceptor to be used. In our experiments, we did laboratory experiments with different conditions, pressure, no pressure, and reservoir, and so on. We see minimal hydrogen consumption. Well, we ran the experiment 3 months, 4 months.
Just when you add some.
Yeah, if you add sulfate, then of course.
Sulfate and
It is-
organic matter.
So you need organics and electron acceptor for things to use it. Otherwise, we did 3 months experiments. You know, we should do maybe a 1 year experiments. I mean, you have students that stay around for much longer, but we didn't see the rates that people are talking. When I go into papers and I look for the rates that people are discussing, it's always in the worst case scenario where you add the sulfate, acetate, formate, yeast extract, stimulating the community, pre-inoculating your microbes. So of course.
It's even difficult to observe microbes under microscope, and it's also difficult to capture them because they are so rare, so specifically shaped by the environment that you have a lot of difficulties to find the right condition to grow them.
And there are evidence that suggests that hydrogen in very high partial pressure is actually inhibiting microbial activity.
Yeah.
So, you know, I'm not on the camp of people that say we should absolutely worry about microbes. I actually think it's going to turn out to be a specific problem of a few set of conditions, which is against my interest because I want everybody to give me money to do it, right? But that's the reality of the data so far. I might be wrong, but data so far suggests this is not going to be a vital issue. It is an issue that we need to figure out together how much of an issue it is, and we're going to need how to manage it in case it turns out to be an issue. I think it's more, for example, I think microbial activity on hydrogen is more important when you think about natural hydrogen, because time scales there become huge. So even small activity by microbes might turn out to be very important for the reservoir and the trapping. Depending on the time scale we're discussing in underground hydrogen storage, months to years, I don't think it's gonna be a huge problem. Hence, 200 years for the different story. Let's try to figure that out. We can discuss more in lunch, or we can discuss more this afternoon.
It's always the same microbiota. They're a generation. How many generations? Yeah, right. Because sometimes the single lineages go together, and so there was sometimes the lineage stopped and then opened up again. In one side.